our first keynote speaker. Frank Hu is the chair of the Department of Nutrition at the Harvard Chan School of Public Health. He's also the Fredrick Professor of Nutrition and Epidemiology at the Harvard School of Public Health, as well as Professor of Medicine at the Harvard School of Medicine, at the Harvard Medical School. He's the individual who made the commitment on the part of Harvard to host this meeting, whether or not the NIH granted us the grant that we ultimately secured to make this available to all of you. That demonstrated his commitment to this conference and this field. We are all deeply grateful to him. Please join me in welcoming my distinguished colleague and dear friend, Frank Hu.
I'm Frank Hu, professor and the chair of Department of Nutrition at Harvard T.H. Chan School of Public Health. The title of my presentation is The Role of Teaching Kitchens in Precision Nutrition Research and Practice. This is the outline of my talk. I will first describe the definition of precision nutrition and the role of omics technologies in precision nutrition research. And then I'll talk about how to translate research findings into practice and the role of teaching kitchens in facilitating precision nutrition research and practice. Globally, we are facing triple threats of obesity, undernutrition, and climate change. Worldwide, we have 2 billion people who are overweight and obese, another 2 billion people who are underweight or undernourished. In the meantime, the world population will grow from 7.5 billion today to 10 billion in 2050. So a major global health challenge in front of us is how to feed 10 billion people a healthy and sustainable diet by 2050. This challenge or problem is exacerbated by the current COVID-19 pandemic, which has disrupted our global food supply chains and worsened health disparity. In the meantime, our food system has contributed greatly to current climate crisis and also global warming. And those environmental changes have increased the likelihood of spread of infections, diseases such as COVID-19. So clearly, the pandemic, our food system, and climate crisis are closely intertwined. Recently, NIH released 2020-2030 strategic plan for nutrition research. This plan is centered around the theme or the vision of precision nutrition. According to this report, precision nutrition is defined as a broad framework that consider a wide range of individual characteristics such as genetics, dietary habits, circadian rhythm, SES, food environment, physical activity, and the microbiome. So this definition is much broader than the traditional concept of personalized nutrition, which is aimed to tailor nutritional strategies to individual characteristics such as genetics and gut microbiome. Nevertheless, the terms precision nutrition and personalized nutrition have been used exchangeably in practice or in research. According to this report, precision nutrition recognizes that humans are inherently different from each other. So when, why, and how we eat is likely as important as what we eat. Precision nutrition has several broad goals, including obtaining more precise measurement of dietary intakes, more precise diagnostics of nutritional status, more accurate prediction of disease risk, identifying more susceptible or responsive individuals to certain dietary exposures, identifying more precise nutritional intervention targets, and the eventual goal is to develop more effective personalized nutrition therapy or dietary recommendations for chronic disease prevention or management. In recent years, metabolomics has become an important and exciting tool in precision nutrition research. This technology can analyze tens, thousands of small molecule metabolites in biospecimens such as blood or urine samples. And those metabolites are mostly derived from metabolism of individual foods or nutrients in our diet. This technology can help us better elucidate the interconnections of metabolic pathways, generate metabolic profiles of organs and tissues, monitor gene-diet interactions, identify nutritional biomarkers, better predict disease risk, and evaluate potential biological functions of genes and microbes. The first utility of metabolomics in precision nutrition research is to develop more objective and more accurate dietary biomarkers. We know that diet is complex and the dynamic exposure with no perfect measurements. Traditional approaches such as 24-hour recalls and FFQs are prone to measurement errors. Omics technologies, especially metabolomics, have the potential to provide objective and more accurate assessment of certain foods and dietary patterns. Traditional nutritional biomarkers include recovery biomarkers and concentration biomarkers. Recovery biomarkers are can directly reflect absolute intake of certain nutrients. For example, double labeled water is a very good biomarker of total energy intake or energy expenditure. 24-hour urine, potassium and sodium can be used to reflect dietary intake of potassium and sodium. 24-hour urine nitrogen can be used to measure protein intake. Concentration biomarkers are correlated with dietary intakes, but cannot estimate absolute intake of those nutrients. Examples include plasma or adipose tissue fatty acids, carotenoids and vitamins, toenail trace elements, and heavy metals. Predictive biomarkers are in between recovery and the concentration biomarkers. One example is the use of urinary sucrose as a biomarker for sugar intake. Although we have a number of validated nutrient biomarkers, there are very few biomarkers for food intakes. In recent years, metabolomics analysis have been used to identify food-based metabolites that can be used as biomarkers of intake of certain foods and beverages, such as consumption of wine, fruits and vegetables, meat, fish, whole grains, coffee, nuts, cocoa, and several dietary patterns, such as vegetarian pattern, Western pattern, and Mediterranean dietary pattern. However, many of those metabolites are not very specific to intake of certain foods or patterns. For example, plasma concentration of TMAO is correlated with red meat intake, but it's also correlated with intake of fish. So high TMAO levels may reflect either high meat diet or high fish diet, depending on the eating habits of the populations. So that's why nutritional metabolomics will be complementary to, rather than a replacement of traditional dietary assessment methods. Reproducible, sensitive, and specific dietary metabolites can be used to validate self-reported measures of diet, calibrate estimates of dietary intakes, provide objective markers of adherence to dietary interventions or certain dietary patterns, and improve predictions of future risk of diet-related diseases and facilitate personalized or precision nutrition. However, we still have a long way to go to identify and validate food-based biomarkers using metabolomics. The second utility of metabolomics analysis is to identify metabolic pathways through network analysis of metabolomics data. And those pathways could be modulated through dietary interventions. Many of you are familiar with the PREDIMED trial conducted in Spain, which is a landmark primary prevention trial of cardiovascular disease with Mediterranean diet supplemented with extra virgin olive oil or nuts. After 5 years of intervention, the intervention diets significantly reduced CVD incidence by 30% compared to the control diet. In the past several years, we have been collaborating with colleagues in Spain to conduct a large-scale metabolomics profiling of PREDIMED participants. The goal of this project is to evaluate the effects of dietary interventions on changes in plasma metabolites and examine whether those changes in metabolomics profiles can explain the benefits of Mediterranean dietary interventions on the risk of type 2 diabetes and cardiovascular disease. This is a summary of our recent findings using data from the PREDIMED trial. Those studies have been published in the last several years. For both type 2 diabetes and CVD, we identified a number of independent, robust predictors, which include branch chain amino acids, glutamine, glutamate, ceramides, and other lipid metabolites, also carnitine metabolites and gut microbiota-related metabolites. Those metabolites significantly predict future risk of diabetes or CVD, independent of traditional risk factors. So those metabolites can be used to improve the prediction of chronic diseases such as diabetes and CVD, and they can also be used as local intervention targets for dietary interventions. In the PREDIMED trial, we also found that Mediterranean diet interventions mitigate the deleterious effects of certain metabolites such as ceramides on CVD risk. The third utility of metabolomics analysis is to elucidate biological mechanisms through multi-omics analysis, which can help us better understand the relationship between diet and disease risk at molecular levels. And they can also help us to quantify individuals' variability in response to certain dietary interventions. One example I want to give is the relationship between coffee consumption and the various health outcomes. Many epidemiological studies have consistently found beneficial effects of regular consumption of coffee on risk of heart disease, diabetes, Parkinson's disease, depression, and some cancers. However, the biological mechanisms underlying those associations are largely unknown. Coffee is a complex beverage which contains not just caffeine, but also a large amount of bioactive compounds such as polyphenols. In this situation, multi-omics analysis can help us better understand potential biological pathways through which coffee consumption improves cardiometabolic risk profiles. For example, we and others have identified a dozen of genetic variants that predict increased or decreased coffee consumption using GWAS analysis. Transcriptomics analysis have found that regular consumption of coffee upregulate anti-inflammatory genes and downregulate pro-inflammatory genes in adipose tissue, liver, and other tissues. Proteomics analysis have found that regular consumption of coffee significantly reduce circulating levels of pro-inflammatory cytokines such as CRP and interleukin-6. In the meantime, metabolomics analysis have identified urinary or plasma metabolites that predict lower risk of type 2 diabetes. So taken together, the analysis from different omics analysis not only improve our understanding of biological pathways or biological mechanisms underlying the association between coffee consumption and the risk of chronic diseases, but also can help us identify individuals who may benefit from increased or decreased coffee consumption. So how can we translate precision nutrition findings into personalized nutrition practice? There are several potential translational approaches. First, dietary advice can be based on individuals' genetic variants that are related to intake and the metabolism of certain foods or nutrients. Secondly, we can measure plasma or urinary metabolites among individuals, which can help us to determine how a person's body responds to certain foods or diet. Thirdly, we can assess gut bacteria species and abundance, which can help us identify specific foods or dietary patterns that can improve blood sugar control or other cardiometabolic risk factors. Finally, machine learning and big data analytics can be used to develop computer algorithms for personalized nutrition practice. Many genetic variants have been found to influence metabolisms of certain nutrients, foods, or eating patterns. Those genetic variants have been associated with certain dietary behaviors or traits such as lactose intolerance, alcohol flush reaction, heavy coffee consumption, strong bitter or sweet taste preference, and increased protein intake. Theoretically, those genetic variants can be used to design more personalized diets or improve individual food choices. However, at this point, there is little evidence that nutrient genomics testing can actually improve individuals' eating behaviors or health outcomes. In recent years, several computer algorithms have been developed for personalized nutrition. In 2015, an Israeli group developed a personalized nutrition predictor, which was designed to reduce postprandial glycemic responses. And this algorithm was found to be as effective as more traditional dietitian counseling in terms of reducing postprandial glucose response. More recently, Tim Spector and colleagues developed a computer algorithm to predict human postprandial responses to foods. So both computer algorithms work very well in their study populations, but it's unclear whether those computer algorithms can be generalized to other populations with different food intakes and eating habits. And also, the predictors in those computer algorithms were not exactly the same. For example, in the Israeli study, microbiome composition was found to be an important determinant of glycemic response, but this was not the case in the UK study. So I think we still have a long way to go before those computer algorithms can be applied widely in clinical settings, because different algorithms developed from different populations and studies may actually lead to inconsistent nutrition recommendations, and different dietary advice may be offered for different health outcomes. Of course, the most important question is whether those computer algorithms can actually improve nutrition quality and health outcomes. At this point, we don't have data to support the clinical utility of those computer algorithms yet. So today, if you ask Alexa, what should I eat? You may not be able to get any meaningful answers. However, this may change in coming years if we have more rigorous conducted studies and more consistent reproducible findings from those studies. In the era of precision medicine, we need to integrate tools and strategies from precision nutrition, public health nutrition, and planetary nutrition. as I described earlier, the reason we want more personalized nutrition is because dietary responses vary substantially across individuals. So there is no one-size-fits-all diet. And also the technological advances in terms of wearable devices, sensors, genetic and microbiome testing, big data, and artificial intelligence have facilitated precision nutrition research and practice. But this doesn't mean we should forget about public health nutrition because personalized approaches benefit only a small segment of the population and may actually widen health disparities. On the other hand, population-based approaches such as policies and regulations are more effective in improving the food environment and population health. In the meantime, we should consider planetary and sustainable nutrition because our current food system has a huge impact on the environment and undermines both human and planetary health. That's why we need a global food system transformation to feed 10 billion people a healthy and sustainable diet by 2050. In this context, teaching kitchens can play a very important role in facilitating research and practice through the inclusion of principles in precision nutrition, public health nutrition, and planetary nutrition, which can help individuals make more sustainable behavior changes through their food choices. Currently, the TKC includes 33 member organizations located in 7 states and several other countries. And those organizations or the network can be leveraged to conduct rigorous intervention studies and test precision nutrition interventions. You can imagine that a multi-site project can be conducted to examine the efficacy of teaching kitchen curriculum with or without personalized nutrition diets on various health outcomes. And the multi-site intervention studies can be also used to assess cooking skills and self-efficacy in preparing individualized meals and evaluate if and how culinary nutrition influences dietary patterns and clinical outcomes. You can also imagine that novel biomarkers of nutrients and food intake derived from precision nutrition research can be incorporated into those intervention studies. And we can also assess the usefulness of precision nutrition computer algorithms as adjuncts to teaching kitchen curriculum. We can also imagine teaching kitchens as translational research labs through which we can teach individuals to adapt dietary guidelines to food, cultural preferences, and individual health outcomes. And then we can also tailor dietary and lifestyle interventions to individuals' genetic background and microbiome composition. Teaching kitchen can be also used as venues for practicums and access to growing research network for the next generation of students and nutritionists, including chefs in training, graduate students, dietetic students, medical, nursing, and other health professionals. It's important to emphasize that even though here we're talking about personalized diet or individualized diet, I think we should also emphasize common nutrition principles described by the dietary guidelines that are very important for everyone to adhere to, besides more personalized or individualized dietary approaches. Teaching kitchens can be also used as a natural experiment or observational studies to examine whether teaching kitchens can actually improve diet quality and reduce health costs and improve health outcomes in organizations or communities that have adopt teaching kitchens. And this kind of settings can allow us to conduct observational analysis on the relationship between diet quality and lifestyle factors and the teaching kitchen practice. So in conclusion, recent advances in genomics and other omics technologies have facilitated precision nutrition research and practice. Precision nutrition has a great potential, but it should be balanced with public health approaches while considering health disparity and environmental sustainability. Teaching kitchens have a lot of potential in terms of facilitating precision nutrition research and practice, but I think that requires enormous amount of investment and also research efforts. I'm going to stop here. Thank you very much for your attention.
Frank, that was fantastic. I thank you. And let me offer two questions, reading from some of the Q&A. One of the questions is really technical. It's about the benefits of coffee, whether it applies only to caffeinated coffee or decaf coffee or tea. And the other question, I'll take the prerogative of asking you myself, now that we know there are hundreds of teaching kitchens, not only in the United States, but around the world, how might you encourage and guide this audience to work together to apply them to precision nutrition research? Back to you.
Okay, thank you, David. Can you hear me well?
Yes.
Okay, so regarding the coffee question, so most of the studies have looked at regular coffee or caffeinated coffee and the risk of chronic diseases such as diabetes, CVD, and several cancers. And the results have been remarkably consistent. And in terms of decaffeinated coffee, a few studies have looked at decaffeinated coffee and the risk of type 2 diabetes and cardiovascular disease. Those studies also showed some benefit, even though the association was a bit weaker than the studies on regular coffee or caffeinated coffee. So at this point, I think the potential benefits of coffee may depend on different health outcomes for diabetes, cardiovascular disease, and some cancers. It's probably not the caffeine that exact the beneficial effect. It's probably other components of coffee, especially polyphenols, chlorogenic acids, and other bioactive compounds, which have anti-inflammatory effects and antioxidant effects. But for mental health outcomes and neurodegenerative outcomes, such as depression, Parkinson's disease, cognitive function, it's likely that caffeine may also have additional effects, beneficial effects over and above the polyphenols and other bioactive compounds in coffee. So the bottom line is that it's a complex story, and that's why we need extensive multi-omics analysis to understand various metabolic or biological pathways underlying those associations.
And my second question, given the trajectory over the next 10 years to make headway in precision nutrition, how do we make use of this growing, rather enormous group of teaching kitchens around the world?
Yeah, I think the teaching kitchens are really valuable settings in which precision nutrition interventions can be conducted. NIH has made precision nutrition as a top priority in the next 10 years. And it's likely that NIH will set aside a large amount of funding for precision nutrition research. The teaching kitchens can help the precision nutrition research and practice in several aspects. One is to recruit participants and conduct intervention studies and incorporate novel biomarkers. And the second area is related to the food as medicine, or food is medicine, which is also a top priority for NIH research in the next 10 years. And that's is extremely important for disease management, especially for people with diabetes, with cardiovascular disease, with cancers. Food is medicine, I think, is really important concept to evaluate in a more rigorous way. And teaching patients, I think, are extremely useful to test the validity and also the efficacy of food is medicine concept.